Welcome to my world. Another day, another time, another period in knowledge acquisition. Knowledge is power, health is wealth. Today, I shall be taking you on the magic bullet. The idea of a magic bullet. That has been mooted since 1917 or thereabout by Paul Ehrlich, and when he accomplished the sulfonamide. After that was the penicillin by Alexandra. And these days, the magic bullet seems to be monoclonal antibodies. Monoclonal antibodies. And I title today's topic, Monoclonal Antibodies Magic Bullet Lab Findings. You will find it interesting. But one thing that may be confusing to some medical lab scientists would say, how does drug, because the idea of the magic bullet is to find a drug that would affect the germ, the causative agent, without having any effect on the human body. And that was how it came about, the sulfonamide, the penicillin, which later were not as effective, though they are still very effective, but not as 100%. Now we have monoclonal antibodies that are being used for therapeutic purposes. But the medical laboratory scientist comes to know more about this through the anti-serras, in the blood grouping serology. So there are so many reagents in the lab these days that are made from monoclonal antibodies that has elevated to enzyme link immunoassay, ELISA test. These are things that I shall try to cover on in the slide section. So stay on. If you like what you've seen, press the like button, subscribe, and be assured of more quality production.
Monoclonal antibodies, laboratory findings. Outline, objective, introduction, production, application and cases, and finally conclusion. The objective, to show the gradual progress of monoclonal antibodies from being used as laboratory reagents or kits to altering its production processes for its application as ubiquitous therapeutic component for human diseases. Introduction. Cloning. The idea of cloning is very popular with dolly sheep, and when you look at that cloning, it is like it came before monoclonal antibody, but in actual fact, monoclonal antibody has been on since the 70s. So, according to WHO, monoclonal antibodies are immunoglobulins derived from monoclonal cell lines, and which have a defined specificity and function. Monoclonal antibodies are laboratory-produced molecules engineered to serve as substitute antibodies that can restore, enhance, modify, or mimic the immune system's attack on cells that aren't wanted, such as cancer cells. So very useful for cancer cells. Therapeutic monoclonal antibodies are in three forms, namely naked antibodies, the naked monoclonal antibody, the conjugated monoclonal antibody, and the bispecific monoclonal antibody. The naked one attacks the antigen directly, while the conjugated helps to link the drug to the specific area that is to be. Then the bispecific monoclonal performs a dual function by simultaneously binding to two different targets. This dual binding capability allows them to bring cancer cells and immune cells together, enhances the immune response against cancer. The picture you've seen is the monoclonal antibody as Y shape, and when it attacks the antigen, it brings signal. This is also to let you know how the production is from the mouse and then mixed together to find the antibody. This specifically shows the rat being injected, and then the antibodies being produced, and the myeloma cells also being produced and mixed together, so that the hybridization, because it's now called hybridoma cells, we are allowed to multiply to give that high quantity of cloned cells, because they are now cloned targeted at specific antigen which they will destroy. An antibody is a Y-shaped protein produced by a type of white blood cells. This is just to let us know the way antibodies are produced. But when you are cloning it, it is now being specific against that targeted cells. And that is the problem, however, is that the monoclonal antibodies were being produced from mouse now have a situation where the body recognizes it as foreign and attacks it. But regardless, once that stimulation has happened, the attraction to that antigen is already done. Then another method of generating monoclonal antibody is by using rats or mouse that are altered so that reaction will not come. Then there is the bacteriophage that acts on E. coli to produce more of this. This is application and case. You'll now find the Antisera blood serology lab uses a monoclonal antibody for its high specificity, and that's what is used to really generate the result of whether you are A positive, B positive, O, AB, and stuff like that. It is also very useful to confirm whether a person is pregnant or not pregnant, because the monoclonal antibody produced against the human chorioniotrophic gonadotrophic hormones is specific. This is a lighter test. You can use it for detecting HIV and other organisms like that. Medicine application and case. From the time the first monoclonal antibody was generated in 1975, the first monoclonal antibody fully licensed in 1986, about 30 monoclonal antibodies are currently approved by FDA for use in humans for treating various diseases and conditions, including cancer, chronic inflammatory disease, transportation, infectious disease, and cardiovascular diseases. That's to let us know that the monoclonal antibodies are becoming very, very useful. And this is another so many cancer treatment. It's used for chronic lymphocytic leukemia. There is also the one used for the bowel cancer and head and neck cancer, and also breast cancer and stomach cancer. In conclusion, monoclonal antibodies presents an attractive option for the development of new kits for hormonal assays, new therapies, and molecular drug target against a wide variety of common diseases due to their specificity and flexibility. As a magic bullet, its production should be encouraged in order to minimize the noise associated with the side effect of COVID-19 vaccine. You can check more of my video on this channel. You're very welcome.